Okay, so today's lecture is Intro to Veterinary Mycology. So what is mycology? Mycology is the study of fungus. Mycology is the study of fungus. Okay, that is called mycology. So what is veterinary mycology? Veterinary mycology is the study of fungus which cause diseases in animals. Very simple. Okay, and the subject is veterinary microbiology, by the way. So let us start with this chapter. Deals with the basic introduction to the fungus, and first is will define the fungus. Will define the fungus. So fungus is eukaryotic, unicellular or multicellular, wait, unicellular, multicellular, non-photosynthetic organisms. Non-photosynthetic organisms. Okay, which cause disease in animals. Okay, so now we will study that. We should know that the cell wall of fungus, cell cell wall of fungus is made up of chitin and polysaccharides. Chitin and polysaccharides. Okay. Okay. So this is the basic point regarding the mycology or fungus. Now there are two forms of fungi. If we talk about the forms of fungi, then there are two forms. Okay, two morphological forms of fungi. So those two morphological forms of fungi are wait. Let me change the color. So two forms of fungus are yeast and mold. Yeast and mold. Hmm. Okay. Now yeast 37 degrees Celsius 24 hours. I will explain what this means. And mold 25 degrees Celsius 7 days. Okay. So what is the meaning of this? This is the incubation temperature for yeast, incubation time for yeast, incubation temperature for mold, incubation time for mold. Okay. Okay. So this is the basic. These are the basic points. Now, if we talk on mold, if we talk on mold, then mold looks something like this, somewhat like this. Somewhat like this. Okay. So this is hyphae, hypha, and its network. So for suppose that this is the network of hyphae. The network of hyphae is called mycelium. Okay, this is called mycelium. Okay. Now, so what is a mycelium? Mycelium is the mass of interlacing hyphae. Okay, mass of interlacing hyphae is called mycelium. Okay. Okay. Now let us talk about reproduction of fungi. So, reproduction of fungi. So, two forms, asexual and sexual. Okay. Now, asexual and sexual both with the formation of spores. Formation of spores. Okay, both with the formation of spores. Okay. Now, respiration of fungus. Respiration of fungus. So there are three forms of respiration: aerobic, facultative anaerobic, strict anaerobic. ठीक है. Okay. Okay, so aerobic 25 degrees Celsius or 37 degrees Celsius. Facultative anaerobic means facultative anaerobic means its preferred site for respiration is, or preferred site for production is in the presence of absence of oxygen, but it can also survive in the presence of oxygen. But strict anaerobe, it is for example ruminal fungi, these cannot survive in the presence of oxygen. Okay. Okay, next is habitats. habitats Next is habitat. So there are three forms of habits or habitats of, or not habitat, sorry, these we'll talk about the habits. So habits of fungi. So there are three forms of habits. First is saprobic or saprophytes, second is parasitic, and third is opportunistic infection causing. Okay, opportunistic infections. Okay, also some fungi produces mycotoxins, so that is a separate chapter, mycotoxin, that we will study later. Okay, now we have studied the main morphological forms of fungus. Now we will study 2 points that are very important in relation to fungus. So terms. Okay, so there are two terms: anamorph anamorph and teleomorph. Anamorph and teleomorph. Okay. Okay, so if I talk about anamorph, then if we talk about anamorph, there it is used to represent that life cycle of fungus when it is, that part of fungus life when it is reproducing asexually. Okay, so anamorph, uh, part of fungal life when it is reproducing asexually. Okay, and teleomorph, part of fungal life when it is reproducing sexually. Okay, these are the two basic terms that are very important with relation to fungus. Now we'll draw the basic groups of fungus. So if we talk about fungus can be divided into lot of groups. So main are 6 groups. So those 6 group of fungus are: first is dermatophytes. Okay. Okay, now if we talk about dermatophytes, then these causes skin infection in animal. Derma means skin and phytes means fungus. So skin infection in both animals and humans. Okay. Okay, after dermatophytes, next is aspergillus. Next is yeast. Okay, yeast. Next is dimorphic fungi. Okay, and next is zygomycetes. And next is mycotoxin producing mycotoxin producing fungi. Okay. Okay, so these are basic groups of fungi. Okay, now let us discuss another point with relation to fungus, that is, let me change the color. this color. And let me just repair my highlight stylus. Okay, so there is a term which is used in reference to fungus that is called Deuteromycetes or fungi imperfecti. Wait, wait, I am not able to write. Deuteromycetes, which is also called fungi imperfecti. Okay, so these include only those includes those fungus which does not have any discovered sexual life cycle. So they have not, we the scientists have not discovered their sexual life cycle yet, so that is why they are kept in Deuteromycetes of fungi imperfecti. Now 7 phylums recognized in kingdom fungi, recognized in kingdom fungi. Okay, kingdom fungi, out of which 3 are of veterinary importance. 3 are of veterinary importance. Okay, so what are those 3 genera of fungi, phylum of fungi which are of veterinary importance? They can be learned by the mnemonics ABZ. ABZ is the form of DNA found in animals and humans. So ABZ is the phylum of the fungi, so which are involved in veterinary sciences. So that is Ascomycota, Basidiomycota, and next is Zygomycota. So these are the 3 phylums which are of veterinary importance. Okay, now let us discuss the incubation temperature for various groups of fungi, the groups of fungi which we discussed just now, so first is name of group of fungi, and second is incubation temperature. Incubation temperature. Okay, name and incubation temperature. So if we talk about dermatophytes, first group is dermatophytes. Then dermatophytes, in dermatophytes the incubation temperature is 25 degrees Celsius. Okay, 25 degrees Celsius. 25 degrees Celsius. Next is Aspergillus. Aspergillus, the incubation temperature is 35 to 37 degrees Celsius. After Aspergillus we have yeast. So any kind of yeast, whether pathogenic or non pathogenic. Pathogenic also 37 degrees Celsius and non pathogenic also 37 degrees Celsius. Okay, next is dimorphic fungi. So now we will not write, okay, dimorphic fungi we can write. Dimorphic fungi which can exist in both morphological form, that is yeast and mold. So yeast and molds. Okay, yeast and mold. So if we talk about yeast, then 37 degrees Celsius again, and mold 25 degrees Celsius, as you have seen in the beginning of our lecture. Okay, and next is Zygomycetes. Then 34 degrees Celsius. Okay, so these are the basic points. Now one more point is left, that is called, that is of very important, that is of high importance. So that point is called mechanism involved in fungal diseases. So mechanisms involved in fungal diseases. Okay, mechanism involved in fungal diseases. So there are three mechanisms which are involved in fungal diseases. Mycosis, mycotoxicosis, and induction of hypersensitivity. Okay, so what is mycosis? Mycosis, that is the tissue, the fungi itself invades the tissue. So fungi itself invades the tissue. Mycotoxicosis, fungi produces toxin which damages the tissue. Okay, and induction of hypersensitivity. So this was a lecture on introduction to veterinary mycology.